up there from the beginning of my work. All I can say is this is out there. It's out there. And I can't find anybody to do the work. I mean, I don't see the work being done. What's obvious to me to do is those research. It's not being done. And to my knowledge, there is nothing that refutes what I'm saying at this point. I guess I should say I hope that it will be, but I can't right now. And so what I'm saying is that the notion of this consideration of this particular condition as affecting just a small segment of the population that we can treat as being off to the side like lepers does not appear to be justifiable to me. Okay, those are the plain facts of the situation. And the last thing that I wanted to mention comes around to this blood thing again. And that is the particular form of great interest has been a dental sample. This is one of many, but a dental sample is of particular interest. And the use of wine, it was an unusual situation how it was done, but the use of wine is producing this filament structure from the teeth, and so far it's actually everybody that's done it to some degree. And if you look at this filament structure that's coming, you see this same kind of thing. Well, the work that I've been doing over the last 6 months or so is it's back in that science fiction world again, because what's happening is I've seen that I'm able to culture these filaments. I'm able to grow them. See, to me, anytime you can culture something or grow it, it opens up a world of possibilities in terms of what now you can do to study that. Now you have something that you could put in the lab, right? You could study it. Controlled environment, classic science stuff. Well, I'm doing work with culturing of these filaments. And there are certain growths that are taking place that, again, are repeating this whole structure that I'm talking about over and over and over. And it becomes even more strange lately in that from these same cultures, there is I can't come to any other conclusion other than it appears there is I always say appear, but there is a blood cell that's appearing from this culture that makes just no sense at all, again, to be there. Again, you don't grow blood cells in a test tube, but it's happening from these filaments. And the part that's different about this test, and we're almost caught up to where I'm ending. I got one more and then I'm done. But when this was happening, where I was seeing this erythrocytic form from these filaments, this is when I sort of on my own, I said, man, I've had enough of this. You know, are these blood cells or not? I mean, they look like it, they measure like it. I've seen them a dozen times over the years, but are they or aren't they? And I really had to try to force the issue beyond what I could do visually. And so this time I really worked hard. It took me at least 2 months or 3 months to get through it. But I went to some what would be called forensic tests, things that would be satisfying the standards of a courtroom to determine whether or not this was blood, whether or not there was hemoglobin in it. And if you look at the work on this particular paper, that particular paper is titled Blood Issues Intensify. Okay? That's the title of that paper. If you look at that, you will see the documenting of 2 forensic level tests, again, done to the best of my ability. One's, was it Castlemeyer and Hemostix. These tests pass for the presence of hemoglobin from these filaments in a way I simply would not expect it, but they pass the test for hemoglobin, presence of hemoglobin. And again, it's out there, the methods are there, you can do it yourself, but this is what I'm seeing. And the most recent one, and this is sort of like just done a couple weeks ago, but the most recent one is not only has there been a culturing process that has been developed that shows these forms developing, but I did something to try and get into the actual fiber to break this apart. And we can call it intuitive hunch. We don't know where things happen sometimes, but sometimes some things happen in unusual and unexpected ways I can't always explain. But I did something to these fibers to see if I could break them down. And the 2 main things that are done there is you have an alkali solution, sodium hydroxide, a strong base, lye, and heat, both of these together. That combination appears to have broken down that filament structure. And there's pictures there and at the end. All I can say is that within the filaments, there are two structures that are showing up. And they again satisfy the conclusion I can make, the best I can, of an erythrocytic form and also that submicron structure. And I wonder, I don't know if I can get to, it would probably be fitting if I can end this by showing you, oh, I know why we're not seeing them. Guess what? You know how I said there were two parts? There's a whole nother set of photographs I never got to, and I'm not going to torture you through them, but I'm going to show the last picture of them, because what's happening here, if I can get to it, I've used the word artificial blood, and I put a question mark by it. The best interpretation that I can make right now is that what is internal to these fibers appears to me to be an artificial or a created form of blood. And the reason for that is twofold. Number one, because of their geometric purity. They're just so perfect if you look at them. I've looked at a lot of blood now, and I see a lot of variations. But these particular cells are just a perfect form. Now, I should have had these pictures going up that whole time I was talking. This is visually leading to what I've been describing all this time, the culturing work, examination of the fibers. I'm after showing you inside. This is the stuff where the cells are coming from a culture form. And this is where it's passing the hemoglobin tests. Those are air bubbles from peroxide. It's a reaction in the Kastle-Meyer test. Hemosticks, positive test. This is the part where I'm talking about the actual fibers of heating them up within a lye solution. And it actually even looks like blood, to tell you the truth. When you're going, okay, what's coming out of this is the end of what I'm talking about. Within those, internal to the filaments, it's like it's too perfect. I don't see blood cells of this pure geometric form all the time. And remember about the two things I keep saying I'm seeing with respect to I'm seeing this internal submicron structure, and this really has my interest up, because we didn't even talk about it, but that's one of the blood variations that I'm seeing in individuals, is this appearance of this structure inside the blood. And so you can call it conjecture or hypothesis, I don't know, but if you read the paper, or it's called artificial blood, and it has a question mark after it, and I'm raising the question is, where does this come from? Also, by the way, this is an extremely hostile environment, right? This is lye and heat. This is not like the normal biological situation. Okay, so what are we dealing with here? And this is actual blood, and it's again that recurring structure inside. Those are blood cells that to me they're anomalous, and that's where the work is at. And you understand, I'm not trying to excite anybody into a state of fear. I'm a researcher. I present the work as fairly and honestly as I can. I do not claim to be right with everything that I do. I do claim to do the best that I can to try to seek the truth. And again, it brings us to considerations that many of us may not be comfortable with, but it is my duty to present them to you. And I'm going to shut up now. And I thank you very much. And we're going to take questions. But I thought maybe we'd take a break for 5 minutes, because you guys have been really patient. If you want, take a break for a couple minutes.